All right, so good afternoon, everyone. I'm almost about to say good evening. I hope everyone is still awake. So first, I would like to introduce myself. So my name is Kalitan, and I'm working in Geneva. So this is the lake that you can see here. And I've been doing my postdoctoral work in the lab of Christian Lucher. And in a couple of weeks, I'll be moving to the north of Switzerland, Basel, to establish my own lab. So I'm here today to present you part of the work I've done in Christian's lab. And the very last part is going to be about unpublished data that I would want to share with you, and I hope to get some feedback. And I also hope to make this last 1 or 2 hours, let's say maybe 1.5 hours, a little bit interactive. Okay, that's good. That's what I'm looking for. All right, so I'm going to basically ask questions, and I hope to get answers. Okay, so the title of the talk, I've changed it at the last minute, just to make it a little bit more precise about what I want to talk and the strategies. So basically, it's still about inhibitory circuits in the reward system, but we're going to be focusing on the ventral tegmental area, and the tools that I have been using are pharmacological and optogenetics. So the reward system, its origin or the core structure is the VTA, ventral tegmental area. I guess you heard about it. Yes, no? What's the major cell population in the VTA? We heard it like half an hour ago. Come on, that's a simple question. It's written right there. This is the VTA. They are GABAergic, but it's not the main one. It's not the most numerous one. Dopamine neurons. I cannot hear you from here. So yeah, the VTA is the core region of the reward system, and it is composed by 70% of dopamine neurons. And these dopamine neurons sends many different projections, for example, to the nucleus accumbens, that is also known as the ventral striatum. The prefrontal cortex, there are many other regions that I did not draw here for simplicity. And so the dopamine is, we're talking about the reward system here. Its other name is called, what is it? What is dopamine for? I'm sure you heard that during the week. Pardon?
As a stimulate
yeah,
it
it's
actually depends to which receptors, which type of dopamine receptors.
Yeah, true. So it's very largely, we're stepping back, it's a neuromodulator. We're talking about the reward system, so it's about pleasure. So it's also known as the pleasure molecule. So whenever you feel good, it's about dopamine. There are other neuromodulators, but we're going to talk about dopamine. And then here we can see that there is also GABAergic neurons. And these GABAergic neurons have local projections onto dopamine neurons, but they send also long-range projections to different regions. And this is what I'm interested in. And today I will just, as you can see, this is a few studies that I highlighted that have done lots of work in dissecting this circuitry. So only few of the projections are highlighted here, but there is connection, see, from the BNST to the VTA. This is the lateral habenula, we just heard about it. So, as I said, the focus is going to be on the VTA. We've just said that the major cell population is the dopamine one, and there is also GABAergic neurons. There is another one, cell population. Which one is it? It's a minor one, glutamate, yeah. We don't know much about them, and it would be interesting to know whether there is a local release of glutamate, the projections, and how they would be involved. But so far, a few labs are working on that. And again, for simplicity, I did not draw them on that second on that schematic. So basically, I'm gonna report about a study where we have used pharmacological tools, and we have shown that this pharmacological drug is able to induce disinhibition of dopamine neurons. So, what does disinhibition mean? Sorry? Opposite. Activation through which mechanism? Is it direct, indirect? Indirect. So basically you release the brake, okay? And you will see that at the behavior level, when you disinhibit dopamine neurons, or with other drugs you can activate directly dopamine neurons, it drives drug reinforcement. So actually that is the behavior that we have looked into. And on the opposite, so we will see that when we have used optogenetic tools and manipulated this synapse in the opposite direction, so we then inhibit dopamine neurons activity at the behavior level, we observed aversion or avoidance, and this we can discuss about these two terms. So for the first part, pharmacological tool that I have been using is the benzodiazepines. Do you know this class of drugs? Yeah, so some names, I mean, some of them are shown here.
So the benzodiazepine.
Yeah, okay. And what are they doing? What are they used for?
Actually, they are allosteric modulators.
So they modulate GABA A receptors. Okay, and what does that do to?
Chloride influx and then hyperpolarization of the cell.
Okay, that is very molecular, cellular based. That's very good. So they are positive allosteric modulators of GABA A receptors. But whenever people, so why would you be prescribed a benzodiazepine? Sorry? Yeah. Some more effects?
And also withdrawal of the
Sorry?
treatment of the withdrawal symptoms.
Withdrawal symptoms. Okay, we're going to come back to that. So anxiety, hypnotic effects, sedative effects, muscle relaxant, epilepsy. So, yeah, hypnotic, sedative, yeah, sleep. That's very good. So, anxiolysis, sedation, muscle relaxation, anticonvulsant effects for epilepsy. Anterograde amnesia. In which case? Retrograde, that's true. Good, you're following. So, in which case?
General anesthesia.
General anesthesia. So, why would you want people to forget about?
Because in the operation they couldn't remember anything when they made.
Exactly. So in some specific surgical conditions, when it's kind of painful, you would want the patient to forget a little bit about the pain. So some benzos are used. And then the last effect is addiction. Now, we can talk about addiction, withdrawal effect, and benzos, but benzos, when they are being prescribed and used for a chronic period of time, they can induce dependence and in some cases addiction. Now they do have also strong withdrawal effect, and so the withdrawal effect is linked to dependence, right? Okay, and actually this study that I conducted was in the context of dependence to that drug, and the question at the beginning was, how do benzodiazepines increase dopamine levels in the brain? So you know that.
One new foundation is that finding is that also they have the analgesic effect.
Analgesic effect, yeah. Through which receptors?
Alpha.
Which alpha?
Alpha subunit of the drug.
So. Alpha.
I don't remember exactly.
Yeah, I'm not sure.
One of them is
so then, since we're talking about that, what's the structure or composition of GABA A receptors? Sorry?
It's five members, subunit.
5 subunits, yeah.
Alpha, 2 alpha, beta,
2 alpha, 2 alpha, 2 beta, and 1 GABA. And then I have asked which alpha. So this means that obviously there is different isoforms. And actually, benzos can induce all these different effects because GABA A receptors can be expressed at different places in the brain, different places in the cells, but they are also composed of different subunit isoforms. So, and depending on which subunit isoforms between the alpha and the gamma subunit, then you can trigger different effects. So, an alpha beta, because this is at the edge of these two subunits that is lying the benzodiazepine binding site. Okay, so but the question was how benzodiazepines are able to increase dopamine levels just as any other addictive drugs. So one experiment that we performed was in vivo recordings in anesthetized animals. So these are single extracellular unit recordings. Do you know this technique? Who is doing this technique? No one is doing in vivo recordings? Yeah?
Actually, I did, but not in the neural cells, but I did in the muscle cells, fibers.
Okay, but this is like in the brain, you have the animal that is under anesthesia, and so maybe you can.
Yeah, my lab, we study visual system plasticity.
Yeah.
So we have neuronics and electronic technology that really give away freely moving animals, and we look at deep potential testing again.
Okay. So at the end, so this case is freely moving animals. You can also do in vivo recordings when the animal is placed under anesthesia. And the way you do it for single cell extracellular unit recordings, you lower down an electrode, a glass electrode, following stereotaxic coordinates, and then while the pipette is lowered down, you can hear the activity of the neurons if you are close enough to a cell. And what we can see here is a histogram that shows the firing rate over time. And here what you have are traces. So the vertical bars, one bar is an action potential. Are you familiar with this kind of data? Yeah? No? Yes? Maybe? Okay. So this is an example trace of the firing rate of a dopamine neuron at baseline. So this is what we have here. This cell fired at around 1.8 hertz. And what we did was to inject midazolam, that is a benzodiazepine which is not or doesn't show much selectivity for one or the other subunit isoform, so it will potentiate any GABA A currents. So this drug is injected through the tail vein and therefore reaches the brain very quickly. And as you can see here, the midazolam does induce the increase of the firing rate of dopamine neurons. And what we did after a few minutes was to inject flumazenil. What is flumazenil? Antagonist of the benzodiazepines. And you can see that the flumazenil is able to quickly reverse the effect of midazolam. So flumazenil is used in clinics when people come and they have an overdose of benzodiazepines. You just give flumazenil and they kind of wake up or recover. So you can see here a nice increase in the firing rate induced by midazolam, and then it comes back to baseline with flumazenil. So if benzodiazepines are supposed to potentiate GABA A receptor, how come we have an increase? Disinhibition. So then what was the next experiment? Oh yeah, sure, where are the GABA A, but you would want to know where or which cells are actually really inhibited by benzodiazepines? Maybe? So then, as I said, the VTA contains 75% of dopamine neurons. There is about 15% of GABAergic neurons, and we recorded from them. and did the same experiment. As you can see here, midazolam induced a decrease in the firing rate of GABAergic neurons. Okay? So yeah, the next question was, where are the GABA A receptors located in VTA cells, or in the VTA? So then we performed immunohistochemistry. So probably I brought this slide too early. So if you were to do immunohistochemistry, you can have a look obviously at the slide. How would you do that? How would you try to answer that question? Where are GABA A receptors?
Can you just make the fluorescence ligand for it, and then we can find the attachment site?
Yes, you could do that. So maybe someone can then describe the image. Yeah. So what we did here was to take a GAD67 GFP mouse, perfuse the mouse, make VTA slices, and then we did a staining for tyrosine hydroxylase, that is the enzyme to produce dopamine. And as you can see, this is an overlay image. I probably should have brought the different panels, but so the red cells are the dopamine neurons, the TH positive neurons, and the green ones are the GFP positive neurons, so presumably the GAD67 expressing cells. And then here, alpha 1, this is one of the subunit isoforms that we use, and we wanted to track it, so we did the staining for it. And what you can and this alpha 1 subunit is visualized in blue, and as you can see here, wherever there is blue, there is green. So because an image is very nice, but by itself doesn't mean much, what we wanted to do was some quantification. And again, you can see the majority of TH positive neurons, a substantial proportion of GAD67 GFP positive cells, so GABA neurons. And then when we looked at the alpha 1 expression, we saw that actually it's preferentially colocalized with GAD67. So the alpha 1 subunit isoform of the GABA A receptors are preferentially expressed in GABA neurons, at least in the VTA. Question? No? So, okay, now we know that alpha 1 containing GABA A receptors are only expressed in GABA neurons. So what would be the next experiment? Yeah. But then if you were to do that, you would think that since the alpha 1 containing receptors are expressed in GABA neurons, you would repeat, let's say, the in vivo experiment. You will provide an alpha 1 selective compound, and then the activity of dopamine neurons would be decreased, and you would disinhibit dopamine neurons, okay? So what does it give you more? So now we know that alpha 1 containing GABA A receptors are expressed exclusively in GABA neurons. So what's the next question? True. Yeah. So how would you do that? She said that she would be interested to look at whether these receptors are located presynaptically or postsynaptically.
You have to check this out.
Yeah, but how you do that? Are you doing this kind of experiments?
It's easy to say.
Yeah. Okay, so we looked at this question doing in vitro electrophysiology. And we recorded miniature currents. And with the frequency and the amplitude of the currents, you can have a hint for the effect of benzos being pre or post. And then you have an idea of whether the receptor is pre or post. This we did. And I would say that there might be some presynaptic receptors, but they're also located postsynaptically. As you can see here, the soma is like really bright with the alpha-1. And it's true, I understand your question because of all these blue fibers. So, but the question that we asked was, okay, it seems that alpha-1 is only expressed in GABA neurons. Are these receptors really doing the job? So when you want to know whether a receptor is the one that does or is at the basis of the effect that you're observing, what do you do? How do you? You can knock it down. Yeah. So we did not knock it down, but we used a transgenic mouse line. Anyone knows about the point mutant GABA A? So can you tell us about it? No. Maybe you can tell us. Yeah. And? Exactly. So is that clear for everybody? Everyone heard that? So there are point mutant transgenic lines where one amino acid residue has been mutated, and it's, for example, in the alpha-1 subunit isoform is the histidine 101 that is mutated into an arginine. And this single mutation is sufficient to abolish the sensitivity to benzodiazepine. And what is very nice with this is that the GABA transmission is intact. So only the sensitivity to benzodiazepine is gone. But of course, any other receptors that do not contain the alpha-1 is still sensitive to benzodiazepine. So then what we did was to repeat the first experiment, the in vivo recordings, in these point mutant mice. And as you can see here, if we were to record from dopamine neurons, the effect induced by midazolam that we observe in the wild-type mice is just gone. And same when we record it from GABAergic neurons. Again, these GABAergic neurons are the cells expressing the alpha-1 containing GABA-A receptors. Okay? So no more potentiation of the alpha-1 containing GABA-A receptors expressed by GABA neurons. Those cells are no more inhibited, so no more disinhibition. Make sense? Okay. So then, if these receptors are sufficient to trigger or at the basis of the disinhibition of dopamine neurons, which means that when benzos are provided to the system, dopamine neurons are disinhibited, dopamine is released in the brain, there is this transient increase of dopamine, and as I said, it's a pleasure molecule, so you feel good, right? So what kind of behavior experiments would you do to test whether the alpha-1 containing GABA-A receptors are important and driving this effect? Place preference, so how would you do that? Yeah, so you would inject benzos, IP, keep the animal in one chamber, do some conditioning sessions, and then on the test day see where the animal likes to spend its time. Yeah, that is possible. We did not do it. We even did not test it, but I think it's more a problem of the way benzos are also used in clinic. And our point was to look at dependence. And most of the time, these are patients that take the drugs, like they eat it, right? It's not really an injection, so we did not want to mimic that. We did something even easier than that. Sorry? Yeah. So how would you check through food if a drug is rewarding or reinforcing, has reinforcing effects?
By measuring the amount of the food which they take and then you give them the gauge and then they after taking this drug, how much it becomes to the food.
Yeah. So we did something similar to that. We did not put the benzos in the food,
And the other one
but
is the water.
but we put it in the drinking solution. So this is the Aversive administration, and it's a very simple task, you know, for the rookies that start behavior, and that's great. That was my case. So we placed 3 animals in a cage. They have free access to food and to 2 drinking solutions, okay? And every day you would come and check how much of food have been eaten, right? Just to see whether they are healthy. You would weigh the animals also to check whether they are healthy. And then you will check how much of this 2 drinking solution has been consumed. We separated the experiments into 3 phases. The first one where the 2 bottles contain water. This is just for the baseline. And then we switched to a sucrose phase. Why would we do that?
Check the taste.
So what about the taste?
No, then you can compare when you have the mixture of the midazolam and sucrose.
But why we would not put midazolam in water?
It's bitter.
Exactly. And actually benzodiazepines have a bitter taste. So that's why we went for the sucrose phase. And then after 2 days of sucrose consumption, then 1 bottle we would add midazolam. And then every day we would weigh the animals, the food, and the content of the 2 drinking solutions. So this is what we observe for the wild-type animals. This is the water phase, the sucrose phase. This is the relative midazolam consumption. Obviously, the intake of the sucrose solution is higher than the water, right? And then, as soon as we have added the midazolam, in one of the 2 bottles, we could observe that there is an increased consumption from the bottle that contains midazolam. So there is a preference for this solution. So what would you expect from the alpha-1 knock-in mice?
Something flat.
Something flat? Do you agree? You don't agree? So what would you expect?
It must have the same pattern with if sucrose or even sucrose consumption is no, the same. I think
The same?
Same with the sucrose. Without
Okay, I'm going to ask you to raise your hands now if you don't want to answer. So who agrees with, what's your name?
Reza.
Can you repeat?
So, okay, this is end of the week, end of the day. I'm going to repeat it. The wild-type animals show a preference for the midazolam-containing solution. What would you expect from the alpha-1 knock-in animals? So those from, when we did the in vivo recordings, inject the compound, there was no effect on the cellular activity. So no release of dopamine. Preference? No preference. Everyone agrees? We indeed observe no preference for either of the bottles. So no preference, no aversion. They just take same amount from the 2 solutions. And this is very nice because we went from in vitro to in vivo. So I said in vitro, I did not show you any in vitro data, but we went from the in vivo recordings to the behavior task and showed really that if there is any effect of benzodiazepine in the brain, at least for anything that is rewarding, This is done through the expression of the alpha-1 containing GABA A receptors that are exclusively expressed by GABAergic neurons. And this, through that mechanism, you can disinhibit dopamine neurons and increase dopamine levels. You have a question?
No.
Sure?
Yeah, maybe later.
Yeah, I hope later. So this is what we just saw, okay? Now, as I said, second part is about manipulating the system in the opposite direction. So here we're not going to inhibit GABAergic neurons, but we're going to excite them and see what happens at the behavior level. So how can we do that? I told you that it was about optogenetic tools. You heard about optogenetics during the week. So someone explains me, what is it about? Can you tell me? No, right in front. You're safe this time.
I don't know.
You don't know about optogenetics, channelrhodopsin. You want to tell us?
Yeah, I don't well, optogenetic, but I read that you infuse the virus.
Yeah.
Channelrhodopsin, so that you can stimulate by light the neuron where you infuse the virus, and so you can excitate them, and you will see the projection, the excitatory projection.
Okay. Anybody using optogenetic tools?
Ah, I see. No.
So you have something to add to that. And don't ask her to repeat. Okay, yeah, she did not hear it, so you can start from the beginning.
We want to excite the GABA and PTA level.
Yeah,
GABA and
exactly.
So we need selective. We need to have a subpopulation defined based on some kind of a promoter that will target the GABA interneurons.
Mm-hmm.
So for that we will use transgenic mice,
Exact.
and we've created combinations under this promoter that will selectively target GABA interneurons.
Yeah.
And then just add the tree-dependent channelrhodopsin. So the channelrhodopsin will be expressed only as both GABAergic interneurons, and then just shine the light into the BTA to excite GABA interneurons.
Yeah, and so how does the channelrhodopsin work?
It's a cation channel.
It's a cation channel that is activatable by blue light.
It's depolarized.
And it depolarizes neurons. Okay. Are there some similar tools that could be used to inhibit neurons? To inhibit neurons? I don't know. Which one? Oh, the dreads. Okay, but yeah, true. Something closer to channelrhodopsin? Halorhodopsin, arch, and the dread is another system. But yeah, works. The dread is actually activated not by light. It's ligand activated. Yeah. So that was all very correct. So what we use is a GAD65 Cree mouse line that we got from Gero Missenbuck. And we would inject animals, so here it's P21, but you can inject them at any age. We used an adeno-associated virus that was serotype 5. Promoter was EF alpha 1, and you need a Cree-dependent system, so basically the channelrhodopsin tagged EYFP sequence is flanked by LOXP sites. And because only GABA neurons would express the Cree recombinase, then only GABA cells would be able to express the channelrhodopsin tag protein. So then, in this case, we waited for 2 days after surgery to do any kind of experiment. 12. 2 weeks, 12 days. So the first thing, obviously, is to make sure that you're actually doing what you expect to do. So first question is, does channelrhodopsin exclusively express in GABAergic neurons? So we took the animals, perfuse them, slice the VTA, and then do the staining for TH to again visualize dopamine neurons. The YFP signal, green signal, is from channel rhodopsin, and we did the alpha-1 staining because with the previous study we have shown that this subunit is only expressing GABAergic neurons, and as you can see here, the channel rhodopsin YFP signal nicely colocalizes with the alpha-1 subunit isoform signal, and you can see on the quantitative pie chart that there is about 90%, 95% of colocalization. So that is very nice. The channel rhodopsin is expressed only in our GABA neurons in the VTA, but we want to know whether it's functional. So whether we are able to depolarize and manipulate the activity of the GABA neurons. So what we did was in vitro recordings. Anybody doing in vitro electrophysiology? Yeah? So you have heard about how we use channel rhodopsin. How would we check here? How did we validate the system?
Based on this drawing,
Yeah?
you're touching a dopamine neuron, you shine the light, you should be able to see a GABA current.
Okay.
Alternatively, you could patch a GABA neuron and see if you can induce activity.
If you can induce what? Activity.
Action potential.
Yeah, action potentials. So what we did in vitro was exactly that. If you patch a dopamine neuron and shine the light, so this was 400 milliseconds, and this is 4 milliseconds. So, and you can see that in each case you induce a current. And this current was blocked by picrotoxin, that is a GABA A receptor antagonist, so it's a GABAergic current. Then we patched GABAergic neurons, and we did shine the light again for 400 milliseconds and for 4 milliseconds. And here it's different. Why is it different? Yeah, so if you want to see whether a cell expresses channel rhodopsin, you have to shine the light for long enough to see this photocurrent or photodesensitizing current, which means that the channel will be on as long as the light is on, right? If you shine the light for that long onto a cell that does not express the channel rhodopsin, you will not see this desensitizing current. This is what we use to really be able to distinguish between a cell that expresses the channel rhodopsin or not. And then obviously we wanted to make sure that there is a functional synapse between the two, so picrotoxin blocking that current was good, but you want also to apply TTX, that is a sodium channel blocker, so blocks synaptic transmission. And again here, when patching dopamine neurons, you erase the current. So that is all very nice. Of course, we know to do in vivo recordings, so we did in vivo recordings. And we showed here that when you record from GABA neurons and shine the light, this increases a lot the activity of the GABA neurons. If you do the same but record from a dopamine neurons, you can see that there is a shutdown or a strong inhibition of the activity of dopamine neurons. Okay? So the system works pretty well, but what do we do with that? Like we showed, just as with benzodiazepines, that there is a functional synapse between GABA and dopamine neurons. So, but at this time we are activating the GABA neurons. What would it do? I mean, what's the point? It's all about whether dopamine is released or not and what it does to the system and what's the readout at the behavior level. Yeah, so why would it be interesting to look at what silencing dopamine neurons would do? If you activate dopamine neurons, you have, and then at the behavior level, we went for this oral self-administration, drug reinforcement, all very nice, the animals ones. I mean, this is pretty rewarding. So if you then, on the opposite, silence dopamine neurons, what would it do? Would it be rewarding? No. So what would it do? Aversion. Okay. So if it's aversion, then what we did was to first look for how the VTA cells respond to an aversive stimulus, like a food shock. So we kept on recording in vivo the activity of VTA cells and provided a quick, significant food shock to the paw of the animal. And we can see that when we record it from dopamine neurons, the food shock triggers an inhibition of the activity. What happens when we record from GABAergic neurons? On the opposite, they are strongly activated. Okay? Yes?
How many neurons do you expect to respond in that pattern? Every GABA and every dopamine neuron?
Do I have that here? So, every GABA neurons that I recorded were excited by the food shock. The majority of dopamine neurons that I recorded were inhibited. And actually, we can have that here. So, about 90% of the dopamine neurons were inhibited, and the rest here, about 12% were non-responsive. However, this brings us to a very interesting point. It's about the heterogeneity of dopamine neurons. They are called dopamine neurons, but it's just like for the GABAergic neurons. There are different kinds of GABAergic neurons, and there are different kinds of dopamine neurons. They are subnuclei. Whether they are located more medially or more laterally, where they would project, whether it's in the neck or the prefrontal cortex, they show differences. And it's true that some, but very few of those that I could record were excited by the food shock. So then, because we have shown that there is this functional connection from GABAergic neurons onto dopamine neurons, and this is GABA A mediated, we wanted to check whether this food shock was also GABA A mediated. So what we did was to infuse bicuculin. So actually, this data is, we had the bicuculin in the recording pipette. So what you do is you lower down the pipette, and then the bicuculin will slowly diffuse in the vicinity of the cell that you're recording. And then after enough baseline activity and enough time for the bicuculin to reach or GABA A receptors from that cell that you're recording from, you apply the food shock. And in half of the cases, we saw that the food shock-induced inhibition is gone. Then the reviewers came back and said, well, can you not have a better way to apply bicuculin so that you have a bigger effect? And obviously, what we did was to infuse the bicuculin. We did not do that at first because obviously bicuculin is a GABA A receptor agonist, and what would you expect if you block? It's an antagonist. If you block GABAergic transmission, yes, but you can induce epileptic seizures. So that's why we did not want to do that experiment at first. Yes. Okay, and when we infused the bicuculin, we had, I think it was about 80% of the dopamine neurons that would not respond anymore to the food shock. So then we said, yeah, maybe this inhibition of dopamine neurons drives aversion. So what behavioral experiments would you conduct? Place conditioning? Yeah, that's exactly what we did. So we use our CPP box, the drug-induced CPP test that we had. Everyone is familiar with this task? Yes. So if I pick up someone, he's going to be able to tell me. Come on, it's the end of the day. Can you explain me? See? Someone else can explain it. Can you explain me? Just start. Someone else will help you.
So you put the
Yeah?
The two compartments are different, the floor is different, the
Yeah.
color is different.
Yeah. Visual cues are different and the flow texture is different.
So he has the possibility to choose what he prefers depending on the stimulus.
Yes. So, you need to have two chambers that are made such that the animal is able to distinguish one chamber to the other one. Flow texture, visual cues, and a spatial organization. But the volumes are equal, right? And these two chambers are separated by a corridor so that the animal, when you start the experiments, make the choice to go to one or the other compartment. But we did not run a standard CPP or CPA task. We made it dynamic, which means that because we were manipulating the activity of the GABAergic neurons with blue light, so what we did was during the conditioning session, you just leave the mouse free to go to any of the compartments. So the pretest day is just like as any CPP, CPA test. So it's just to see whether the animal has a natural preference or aversion for one of the two chambers. Then we run two conditioning sessions where only one chamber is associated with turning on the light and therefore activating GABA neurons in the VTA. And then on the test day, okay, video of how it goes during the test session. And you can see here that the light is on. So you can see blue light, and the light will be on continuously until the mouse exits that chamber. Okay? And then on the test day, there is no blue light, no more stimulation of VTA GABA neurons. And we check different parameters, and obviously one is the time that is spent in the chamber 1 versus chamber B, A and B. So these are the data. As a group control, we use obviously the GAD-CRE negative animals. They were all injected with the same virus. And then what you can we have plotted here is the time spent in the light pair chamber minus the time spent in the unconditioning chamber for the pretest and on the test day. And you can see that there is nothing happening with the GAD-CRE negative animals. But when we observe how the GAD-CRE positive animals behaved, they showed a strong preference for the light pair chamber. And this is very clear on the tracking plot. This is an example trace. Yes?
Which simulation? What simulation parameters?
Yeah, so here we did continuous light stimulation, which now I think it's a very bad idea. But lately we repeated that experiment with 20 Hertz, and we obtained the same results.
Do GABAergic neurons have a phasic activity?
All I can say is based on the recordings that I have done under anesthesia, and obviously this changes the pattern of activity. Most of them were pretty regular, but they fire to 10 Hertz. In vivo, when there's no anesthesia, they are fast spikers. So they can reach even 100 Hertz. So for the phasic, like you would say, a phase of high frequency and then lower and then higher, this is what you're saying. And therefore the continuous light stimulation would not be appropriate, possibly. So, but when we repeated that experiment with the 20 Hertz stimulation, we obtained the same results. So, what I showed is that on the opposite to the benzos, if you're able to selectively activate the activity of GABA neurons, you then inhibit dopamine neurons activity, and you decrease obviously the levels of dopamine in the brain, and this drives behavioral aversion or avoidance. So what experiments would you do now to really show that? I'm keeping saying that I manipulate GABA neurons activity, but at the end what is important is the level of dopamine. So what experiments would you do to really show that It's really the activity of dopamine neurons that matters. So who talked first? That was you? Ah, yeah. What did you say?
Inhibit dopaminergic neurons.
Exactly. And we did that. So we use a Th3 mouse line, infect the VTA with a virus that allows the expression of halorhodopsin and run the exact same behavioral task. And we observe also behavioral aversion. So that is pretty nice and pretty consistent.